How's it going, everyone? It's Chow here, and today we are going to briefly discuss the polymerase chain reaction, also known as PCR. So let's get started. Okay, so PCR is a pretty straightforward process, so I'm going to keep this very quick, but it's an in vitro process, which means it's in a test tube, not in an actual model organism, where you're going to run a series of cyclic processes of denaturing, annealing, and extension to create multiple copies of a gene of interest. So you basically make duplicates each time, so you exponentially get more copies. Afterwards, you can take those DNA strands and insert them into plasmids to create that recombinant DNA for biotechnology and basically be cloned into host cells, or you can simply increase the product to allow for better visualization processes and purposes. So aside from the obvious PCR machine, you need a couple of other things, including a DNA polymerase to basically, do the whole replication thing. You need a template strand, again, to basically help out with the whole replication thing. You need a primer, so if you think back to our discussion of DNA replication, you'd know that DNA polymerase can't start replicating the DNA without a primer. So you need to prime that process by putting in a primer within the polymerase chain reaction itself. And finally, you need your deoxynucleotide triphosphates, which are basically your bases. In this case, for DNA, it's your adenines, your cytosines, your guanines, and your thiamines, and you need to put in those DNTPs into the mix to allow for basically extension of your DNA strand. So that's basically the basic important gist and basically materials you need to get your PCR reaction started. Now, what's going to be important, of course, is this cyclic process. So PCR is basically a process of nothing more than denaturing, annealing, and extending. So in order to start PCR, you need to denature your DNA strands. Remember, there are those hydrogen bonds that are going to be pulling those strands together and preventing them from coming apart, and other things like base stacking interactions. But the thing is, you have hydrogen bonds. To break those hydrogen bonds, normally in the cell, you use something like helicase. But in the PCR, you don't use a helicase, you just increase the temperature. So you raise the temperature to above 90 degrees Celsius. Here it says 95, so we'll just go with 95 degrees Celsius. But you raise the temperature to 95 degrees Celsius, and that actually denatures the DNA and breaks those hydrogen bond interactions. After you denature the DNA, you need the annealing of the primers. So remember, you need primers to basically get things started. So in order to anneal the primers, you need to lower the temperature to about 55 to 60 degrees. And when the temperature is about 55 to 60 degrees, the primers will basically anneal. So these are the primers. They will anneal to those strands. After the primers are annealed, you're ready to extend the strand. And that's where you use basically another temperature shift by increasing the temperature from 55 to 60 degrees all the way to about 72 degrees. And at 72 degrees, this allows a DNA polymerase called Tack Polymerase to extend the strand and basically make those two DNA strands, each being a double strand. So you start off with one single double-stranded DNA. Again, you raise the temperature to above 90 degrees to denature it, break those hydrogen bonds, and then you basically lower it to about 55 to 60 degrees to let those primers anneal. And then once those primers anneal, you increase the temperature to about 72 degrees to allow that DNA polymerase to extend it in their respective directions, again from 5' to 3' in terms of synthesis. And finally, you run this process about 25 to 35 different cycles, and that actually allows you to amplify that DNA from just that single strand to a multitude of strands, which can allow you for recombinant DNA technology or just simply for visualization purposes. So we use a DNA polymerase called Tack Polymerase because it generally is a little bit more capable of withstanding high denaturation temperature. So you're putting it in about 90 degree temperature at one time. So Tack Polymerase does a better job of staying folded and actually active when it needs to be and not being denatured itself by raising the temperature. Obviously, in terms of reactivity, Tack polymerase is most reactive and capable at 72 degrees Celsius, but because of the relative stability of Tack polymerase, by raising the temperature to 95 degrees Celsius, you don't actually damage the protein itself. So that's why we use Tack polymerase, but it in itself is a DNA polymerase. Well, that's basically it. Well, I don't really have anything else to say for PCR. Let me check my notes just to make sure. Okay, no, I don't really have anything else for PCR. So this is a basic rundown of the polymerase chain reaction, also known as PCR. And as always, best of luck studying, and I will see you in the next video.